Hello and welcome to Midnight Physics Channel. Tonight we embark on a gentle journey through the hidden landscapes of the human mind. A journey filled with wonder, discovery, and the quiet mysteries that make us who we are. As you settle into this moment, allow yourself to relax completely. There is nowhere you need to be, nothing you need to do except simply be present for these extraordinary truths about the mind. We will explore the fascinating ways your brain works while you rest, how memories form and fade, and the beautiful mechanisms that connect us all. Some facts will surprise you, others will feel like old friends, confirming what you have always sensed to be true. Each one opens a window into the remarkable architecture of human experience. So take a deep breath, let your shoulders drop, and prepare to discover the soothing science of the mind. Even as your body rests, your mind continues its tireless work. During sleep, your brain engages in a process scientists call memory consolidation. This remarkable phenomenon was first seriously investigated in 1924 by psychologists John Jenkins and Carl Dallenbach, who discovered that people who slept immediately after learning retained information far better than those who stayed awake. What happens during these quiet hours is nothing short of extraordinary. Your hippocampus, that seahorse-shaped structure deep within your brain, begins replaying the day's experiences. Like a diligent librarian organizing books, it sorts through everything you encountered, deciding what deserves permanent storage and what can be released. During slow-wave sleep, the deepest stage of rest, your brain waves synchronize in slow, powerful rhythms. These oscillations facilitate a conversation between your hippocampus and your neocortex, the outer layer where long-term memories reside. The hippocampus sends memory traces outward, and the neocortex integrates them into your existing knowledge networks. This process transforms raw experiences into lasting memories. A conversation you had, a skill you practiced, a moment that moved you. All of these become woven into the fabric of who you are while you sleep. Without this nightly maintenance, learning would be nearly impossible. Not all memories are created equal. The moments that carry emotional weight, whether joy, fear, surprise, or sadness, etch themselves far more deeply into our minds than neutral events. This phenomenon has been extensively documented by cognitive neuroscientists studying the relationship between emotion and memory. At the center of this process lies the amygdala, an almond-shaped cluster of neurons buried deep within each temporal lobe. When you experience something emotionally significant, your amygdala activates intensely, releasing stress hormones and neurotransmitters that signal to other brain regions. This matters. Remember this. The amygdala does not work alone. It communicates with the hippocampus, enhancing the encoding of emotional experiences. Research by Elizabeth Kensinger at Boston College has shown that emotional arousal leads to what she calls focal enhancement. Certain details of emotional events are remembered with striking clarity, while peripheral information may fade. This is why you can recall exactly where you were when you heard surprising news, yet struggle to remember what you had for lunch last Tuesday. The emotional significance of an event determines how thoroughly your brain processes and stores it. Interestingly, this emotional tagging evolved for survival. Our ancestors needed to remember which situations posed danger and which offered reward. A close encounter with a predator, the location of abundant food, the trustworthiness of others, these memories carried life or death importance. Today, this same mechanism shapes our personal histories. Your most vivid memories likely center on emotionally charged moments, falling in love, experiencing loss, achieving long-sought goals, or facing unexpected challenges. the relationship between sleep and memory extends far beyond simple consolidation. Research has revealed that sleep actually transforms memories, extracting patterns and insights that were not apparent during waking hours. Scientists call this process abstraction or schema formation. Your sleeping brain does not merely preserve individual memories, it finds connections between them, identifying underlying rules and relationships. This happens primarily during rapid eye movement sleep, that stage characterized by vivid dreams and heightened brain activity. A fascinating study published in the Journal of Neuroscience demonstrated this beautifully. Researchers taught participants a complex sequence of movements. Those who slept after learning showed not just better retention, but a deeper understanding of the underlying structure of the task. They had extracted the gist of what they learned, making them more adaptable when faced with variations. This transformation explains why solutions to difficult problems often arrive in the morning. Thomas Edison famously used this phenomenon deliberately. He would hold metal balls while napping in his chair. As he drifted toward deeper sleep, his hand would relax, the balls would clatter to the floor, and he would wake with fresh insights into whatever problem he had been pondering. The phenomenon is not limited to famous inventors. Countless people have experienced the sleeping on it effect. A thorny work problem that seemed insurmountable before bed suddenly has an obvious solution in the morning. A creative project that felt stuck begins to flow freely after a good night's rest. This happens because sleep allows your brain to make connections it could not forge during waking consciousness. Free from the constraints of focused attention and logical processing, your sleeping mind wanders through vast networks of associated memories, finding unexpected links and novel combinations. Perhaps one of the most beautiful discoveries in neuroscience is neuroplasticity, the brain's ability to reorganize itself by forming new neural connections throughout life. For much of the 20th century, scientists believed the adult brain was fixed and unchangeable. We now know this is profoundly wrong. Every time you learn something new, practice a skill, or even think a thought repeatedly, you alter the physical structure of your brain. New synapses form, existing connections strengthen or weaken, and entire neural networks reorganize themselves. This was first compellingly demonstrated in 1964 by Marion Diamond at the University of California, Berkeley. She showed that rats raised in enriched environments with toys, mazes, and social interaction developed thicker cortexes and more neural connections than rats in standard cages. The brain, it turned out, was not merely shaped by genes but actively molded by experience. Donald Hibb, a pioneering neuropsychologist, articulated the fundamental principle that governs this process. Neurons that fire together, wire together. When you repeatedly activate a particular pattern of neurons, say, practicing piano scales or memorizing vocabulary, the synaptic connections between those neurons strengthen. The pathway becomes more efficient, requiring less effort to activate. This is why practice makes permanent. A skill that initially demands intense concentration eventually becomes automatic. The neural pathway has been reinforced through repetition until it fires almost effortlessly. Modern brain imaging studies have made these changes visible. Researchers using magnetic resonance imaging can literally watch the brain reorganize itself. London taxi drivers who must memorize thousands of streets show enlarged hippocampi, the brain region responsible for spatial memory. Musicians who practice extensively develop expanded areas controlling finger movements. Even something as simple as learning to juggle produces measurable changes in brain structure within weeks. A study published in Nature found that adults who practiced juggling for 3 months showed increased grey matter in regions responsible for visual motion processing. Understanding neuroplasticity has profound implications. It means that you are not locked into fixed patterns of thinking or behaving. With deliberate practice and attention, you can rewire your brain's habitual responses. The brain you will wake with tomorrow will not be quite the same as the one you have today. In recent decades, psychologists have discovered something remarkable. The simple act of feeling grateful produces measurable physiological benefits. This is not merely about feeling good in the moment. Gratitude actually alters your body's stress response. Research led by Robert Emmons at the University of California, Davis, has shown that people who regularly practice gratitude experience lower levels of cortisol, the primary stress hormone. One study found that grateful individuals had approximately 23% lower cortisol levels than those who did not practice gratitude. Cortisol is released by your adrenal glands when you perceive stress or threat. In the short term, it serves important functions, increasing glucose availability for quick energy, enhancing memory formation for stressful events, and suppressing non-essential functions. But chronic elevation of cortisol wreaks havoc on your body. Persistently high cortisol levels contribute to weight gain, particularly around the abdomen. They suppress immune function, making you more vulnerable to illness. They interfere with sleep, impair memory, and increase risk of cardiovascular disease. Gratitude appears to counteract these effects. When you focus on what you appreciate, your parasympathetic nervous system activates. This is the rest and digest branch of your autonomic nervous system, responsible for calming your body after stress. Your heart rate slows and becomes more variable, a sign of healthy nervous system flexibility. Your breathing deepens, muscle tension releases, blood pressure decreases. These physical changes occur automatically when you genuinely feel grateful. The mechanism involves several neurotransmitters. Gratitude increases production of dopamine and serotonin, chemicals that elevate mood and create feelings of contentment. It also stimulates release of oxytocin, sometimes called the bonding hormone, which promotes feelings of connection and trust. Studies using functional magnetic resonance imaging have identified specific brain regions activated by gratitude. The medial prefrontal cortex, involved in emotional regulation and decision-making, shows increased activity. This region also connects to parts of the brain that modulate the amygdala, your threat detection center. The practice need not be elaborate. Simply identifying 3 things you appreciate before sleep has been shown to improve sleep quality and next-day mood. Some people keep gratitude journals, recording moments of appreciation daily. Others make it a regular part of conversations, sharing what they are thankful for. What matters is consistency and authenticity. Rote repetition provides little benefit, but genuinely connecting with feelings of appreciation, really allowing yourself to feel grateful, produces profound effects. Among the most surprising findings in health psychology is that simply writing about your feelings can produce measurable improvements in well-being. This phenomenon has been studied extensively by James Pennebaker at the University of Texas and replicated by researchers worldwide. In a typical study, participants write about a traumatic or emotionally difficult experience for 15 to 20 minutes on 3 to 5 consecutive days. That is all, no therapy, no discussion, just private writing. Yet the effects are striking. People who engage in this expressive writing show improved immune function, with higher levels of infection-fighting white blood cells. They require fewer visits to physicians in the months following the intervention. They report better sleep, reduced symptoms of depression and anxiety, and improved mood. The benefits extend beyond subjective reports. Wound healing accelerates in people who write about emotional experiences. Blood pressure decreases. Markers of inflammation in the blood decline. These are tangible, physiological changes arising from what might seem like a purely psychological intervention. Why does this work? Pennebaker proposes several mechanisms. First, traumatic experiences often remain somewhat disorganized in memory. They exist as fragments, images, sensations, emotions, rather than coherent narratives. This fragmentation requires ongoing cognitive effort to maintain, creating a background burden of mental processing. Writing forces you to construct a narrative, to organize these fragments into a coherent story with a beginning, middle, and end. This organization reduces the cognitive load. The experience becomes something you can put aside rather than something your mind must continuously work to contain. Second, suppressing thoughts and feelings about difficult experiences requires effort. This suppression is never complete. The unwanted thoughts leak through, intruding at unexpected moments. The mental energy spent on suppression becomes unavailable for other tasks, creating a subtle but persistent drain on cognitive resources. Writing provides an opportunity to stop suppressing, to acknowledge the experience fully. Paradoxically, this acknowledgement often reduces the frequency of intrusive thoughts. Once expressed, the experience loses some of its urgency. Third, the act of translating feelings into language engages different neural networks than simply experiencing emotion. Language processing involves the left hemisphere's analytical capacities. By putting feelings into words, you activate these cognitive systems, which can help regulate the more primitive emotional responses generated by structures like the amygdala. The writing need not be polished or grammatically correct. Stream of consciousness works perfectly well. What matters is honest engagement with the emotional content, not literary quality. Interestingly, the benefits appear to be dose-dependent. Writing for 3 to 5 sessions produces reliable effects. Single sessions help, but less dramatically. Very prolonged writing, more than a week or two, does not appear to add additional benefit. There seems to be an optimal window for this intervention. Human touch is not merely comforting, it has measurable analgesic effects. Research has shown that holding someone's hand during a painful experience can significantly reduce both the subjective experience of pain and objective physiological markers of distress. This phenomenon was elegantly demonstrated in studies by Pavel Goldstein at the University of Colorado. Couples were brought into the laboratory, and one partner was subjected to mild heat pain while brain activity was monitored. When the other partner held their hand, pain ratings decreased substantially compared to when they faced the pain alone. Even more remarkably, the partner's brain waves began to synchronize when holding hands. Their neural oscillations aligned in patterns called interpersonal synchronization. The degree of synchronization predicted the amount of pain relief. Greater neural coupling between partners produced greater analgesic effects. Several mechanisms contribute to touch-based pain relief. First, touch activates sensory pathways in the spinal cord and brain that compete with pain signals. The nervous system has limited bandwidth for processing sensory information. Strong tactile input can partially block pain signals from reaching conscious awareness, AII, phenomenon called gate control theory, first proposed by Ronald Melzack and Patrick Wall in 1965. Second, comforting touch triggers release of oxytocin, a neuropeptide with numerous effects on social bonding and stress reduction. Oxytocin has direct analgesic properties, acting on pain pathways in the brain and spinal cord. It also reduces activity in the amygdala, dampening the emotional distress that often accompanies pain. Third, touch from a trusted person activates the brain's reward circuitry, releasing endorphins and enkephalins, the body's natural opioid compounds. These molecules bind to opioid receptors throughout the nervous system, creating genuine pain relief. The quality of touch matters significantly. Gentle, affectionate touch produces stronger effects than neutral or clinical touch. Touch from loved ones works better than touch from strangers, though even stranger contact provides some benefit. The intention behind the touch appears to matter. Touch given with compassionate intent produces different neural responses than identical physical contact given without emotional connection. Cultural factors influence both the frequency of touch and its effects. Some cultures maintain extensive physical contact, while others favor more distance. Yet across cultures, appropriate comforting touch appears to activate similar neural mechanisms. This has practical implications for pain management. In medical settings, having a support person hold a patient's hand during painful procedures can reduce both pain and anxiety. For chronic pain conditions, regular affectionate touch from partners or massage therapists may provide meaningful relief alongside other treatments. Your sense of smell has a unique relationship with memory and emotion, different from all other senses. Olfactory information travels directly to the limbic system, the brain's emotional center, bypassing the thalamus, which processes most other sensory input. This direct connection explains why a particular fragrance can instantly transport you to a specific moment in your past with startling vividness. The smell of fresh-cut grass might return you to childhood summers. A particular perfume might evoke a person you have not seen in years. The scent of cinnamon might recreate holidays from long ago. This phenomenon is called the Proustian effect, named after French author Marcel Proust, who famously described how the taste and smell of a Madeleine cookie dipped in tea triggered a flood of childhood memories. In his novel, In Search of Lost Time, Proust eloquently captured how scent-linked memories arrive with emotional intensity rarely matched by other types of recall. Rachel Hertz, a psychologist at Brown University, has studied olfactory memory extensively. Her research shows that memories triggered by smell are not necessarily more accurate than those triggered by other cues, but they feel more emotional and evocative. People report stronger feelings of being brought back to the original moment when memory is cued by scent. The olfactory bulb, which processes smell information, has direct connections to two critical brain areas: the amygdala, which handles emotion, and the hippocampus, which manages memory formation. This anatomical arrangement means that scents become intimately bound with the emotional context in which they are first encountered. This explains why smell-based memory associations form so powerfully. A scent experienced during an emotionally significant moment becomes neurologically linked with that emotion. Encountering the scent again reactivates not just the memory but the emotional state that accompanied it. The effect works for both positive and negative associations. The smell of hospital disinfectant might trigger anxiety in someone who experienced trauma in a medical setting. The scent of a loved one's cologne might bring comfort and warmth. These responses occur automatically, often before conscious recognition of what triggered them. Interestingly, olfactory memory appears more resistant to forgetting than visual or auditory memory. Scent associations formed in childhood can remain intact decades later, even when other details of the experience have faded. This durability may relate to the direct connection between olfactory processing and memory consolidation systems. Regular meditation practice does not just feel calming, it physically alters brain architecture. This has been demonstrated through numerous neuroimaging studies over the past 2 decades. Sarah Lazar, a neuroscientist at Massachusetts General Hospital, conducted groundbreaking research using magnetic resonance imaging to examine the brains of long-term meditation practitioners. She found several consistent structural differences compared to non-meditators. The prefrontal cortex, involved in planning, decision-making, and emotional regulation, showed increased thickness in meditators. This region typically thins with age, but meditation appeared to slow or partially reverse this atrophy. 50-year-old meditators had prefrontal cortex thickness comparable to 25-year-olds. The hippocampus, critical for learning and memory, showed increased gray matter density in people who completed an 8-week mindfulness meditation program. This structural change correlated with self-reported improvements in psychological well-being. Perhaps most striking, the amygdala, your threat detection center, showed decreased volume following meditation training. This shrinkage accompanied reductions in perceived stress and anxiety. A smaller, less reactive amygdala means a calmer baseline emotional state and less intense reactions to potential stressors. These changes do not require decades of practice. The 8-week mindfulness-based stress reduction program, developed by John Kabat-Zinn, produces measurable brain changes in novices. Participants who meditate for just 30 minutes daily show structural alterations within weeks. The mechanisms underlying these changes relate to neuroplasticity. Meditation involves sustained attention, emotional regulation, and interoception, awareness of internal bodily states. Repeatedly engaging these mental processes strengthens the associated neural networks, just as physical exercise strengthens muscles. Different meditation styles appear to engage different brain networks. Focused attention meditation, which involves concentrating on a single object like breath, strengthens networks involved in attention control. Open monitoring meditation, which involves observing thoughts and sensations without judgment, enhances networks supporting awareness and emotional regulation. Loving-kindness meditation, which cultivates feelings of compassion toward oneself and others, activates empathy networks including the insular and anterior cingulate cortex. Regular practice appears to increase baseline activity in these regions, making compassionate responses more automatic. The benefits extend beyond brain structure to function. Meditators show improved attention span, better emotional regulation, reduced mind wandering, and enhanced ability to stay present. They report lower levels of anxiety, depression, and chronic pain. These are not placebo effects. The structural and functional changes are objectively measurable. The brain adapts to meditation practice just as it adapts to any repeated activity, building specialized capacity for the mental states cultivated during practice. In the early 1990s, neuroscientists in Italy made an accidental discovery that revolutionized our understanding of social cognition. Giacomo Rizzolatti and his colleagues were studying motor neurons in macaque monkeys when they noticed something extraordinary. Certain neurons fired not only when a monkey performed an action, but also when the monkey observed another individual performing that same action. These mirror neurons appeared to simulate observed actions in the observer's own motor system. Subsequent research has identified mirror neuron systems in humans, located primarily in the premotor cortex and inferior parietal lobule. These networks activate when you watch someone else perform an action, as if you were performing that action yourself. This neural mirroring extends beyond simple movements to emotions. When you observe someone experiencing pain, disgust, or joy, your brain activates many of the same regions that would fire if you were having that experience directly. You simulate their inner state. This provides a neural basis for empathy, the ability to understand and share the feelings of another. Your brain quite literally resonates with the experiences of others, creating a shared neural representation. Christian Keizers at the Netherlands Institute for Neuroscience has shown that mirror systems activate even when observing emotional expressions. Seeing someone smile activates your own facial muscles slightly, preparing the same expression. Witnessing someone in pain activates your own pain networks. This automatic simulation helps us understand intentions and predict behavior. By running a simulation of what someone is doing, your brain can infer why they are doing it and what they might do next. This capacity is fundamental to social interaction and communication. The discovery of mirror neurons has profound implications. It suggests that empathy is not merely learned, but built into our neural architecture. We are wired to resonate with others, to feel with them, to understand them from the inside out. Interestingly, mirror neuron responses vary between individuals. People with high empathy show stronger mirror neuron activation when observing others' emotions. Those with conditions that impair social understanding, such as autism spectrum disorders, sometimes show reduced mirror neuron activity, though this finding remains controversial and actively researched. The system can be trained. Practices that enhance empathy, such as loving-kindness meditation, appear to strengthen mirror neuron responses. With repeated cultivation of compassionate attention to others' experiences, the brain becomes more responsive to their emotional states. We have all experienced it. Someone yawns, and within moments, we find ourselves yawning too. This contagious yawning is not mere coincidence. It reflects deep-seated neural mechanisms related to empathy and social bonding. Contagious yawning appears unique to species with sophisticated social cognition. Humans, chimpanzees, and bonobos all show the phenomenon. Dogs can catch yawns from humans, and interestingly, they are more likely to yawn when watching their owner yawn than when watching a stranger. The effect strengthens with social closeness. You are significantly more likely to catch a yawn from a close friend or family member than from an acquaintance or stranger. Studies have found that yawn contagion occurs about twice as frequently between closely bonded individuals. Children begin showing contagious yawning around age 4 or 5, coinciding with the development of theory of mind, the ability to understand that others have mental states separate from one's own. Before this developmental milestone, observing yawns does not trigger yawning. Individuals with autism spectrum disorders who often struggle with typical social cognition and empathy show reduced susceptibility to contagious yawning. This correlation has led researchers to propose that yawn contagion might serve as a behavioral marker of empathic capacity. The neural mechanisms involve the same mirror neuron systems that allow us to simulate others' experiences. When you see someone yawn, your brain automatically begins simulating that action. If the simulation reaches sufficient strength, it triggers an actual yawn. Regions involved in self-awareness and social cognition, particularly the posterior cingulate cortex and precuneus, show activation during contagious yawning. These areas are part of the default mode network, which engages during rest and when thinking about oneself and others. Interestingly, even reading about yawning or thinking about yawning can trigger the response. The mental simulation alone is sometimes sufficient. This demonstrates how powerfully our internal representations can influence our physiology. Why yawning itself occurs remains somewhat mysterious. Yawning increases blood flow to the brain, stretches the jaw and facial muscles, and may help regulate brain temperature. The original yawn likely serves physiological functions related to arousal and attention. But the contagious nature appears to serve social functions, synchronizing group states, promoting social bonding, and facilitating coordination. When members of a group yawn together, their physiological and mental states align, potentially preparing them for collective action. Dreams have fascinated humans throughout history, inspiring countless theories about their purpose and meaning. Modern neuroscience has revealed that dreams, particularly those during rapid eye movement sleep, serve important functions in emotional regulation. Matthew Walker at the University of California, Berkeley, has proposed that rapid eye movement sleep provides overnight therapy, a time when the brain processes emotional experiences from waking life, while stress neurochemistry is suppressed. During rapid eye movement sleep, norepinephrine, a stress-related neurotransmitter, drops to its lowest levels anywhere in the sleep-wake cycle. This creates a unique neurochemical environment where emotional memories can be reactivated and processed without the full physiological stress response. The brain revisits difficult experiences, but in a safer context, gradually stripping away some of their emotional charge while preserving the factual content. Research supports this theory. People deprived specifically of rapid eye movement sleep show impaired emotional regulation the next day. They perceive neutral faces as more threatening and have difficulty suppressing emotional responses. One night of full rapid eye movement sleep appears to recalibrate emotional reactivity. The content of dreams often reflects emotional concerns from waking life, though usually in distorted symbolic forms. Rosalind Cartwright, a prominent dream researcher, found that people going through divorce have dreams filled with themes of loss, conflict, and uncertainty. As they adjust to their new circumstances, dream content gradually shifts. The bizarre, illogical nature of dreams may serve a purpose. By recombining elements from different experiences in unusual ways, the dreaming brain makes connections that would not occur during logical waking thought. This creative recombination might help extract general principles from specific experiences. Some researchers propose that dreams function as a kind of threat simulation. Our ancestors faced numerous physical dangers, and dreaming about threatening scenarios, being chased, falling, facing dangerous animals, allowed mental rehearsal of survival responses in a safe context. Modern dreams continue this pattern, though the threats have changed. Today, we dream about social embarrassment, professional failures, and relationship conflicts, the threats relevant to contemporary life. These dream simulations may help prepare responses to potential waking challenges. Not all dreams carry emotional content. Some appear to be random neural activity, the brain's background processes generating fragmentary images and narratives without deeper meaning. But dreams with strong emotional themes, particularly recurring dreams, often reflect unresolved psychological concerns. For most of the 20th century, neuroscientists believed that humans are born with all the neurons they will ever have. Neurogenesis, the birth of new neurons, was thought to occur only during early development. We now know this is incorrect. The adult human brain continues generating new neurons throughout life, particularly in the hippocampus. This discovery overturned decades of scientific orthodoxy and opened new possibilities for understanding learning, memory, and potential treatments for neurological conditions. The evidence came gradually. In the 1960s, Joseph Altman found hints of adult neurogenesis in rats, but the scientific community dismissed his findings. In the 1990s, Elizabeth Gould at Princeton demonstrated neurogenesis in adult monkeys. In 1998, Peter Erickson showed definitively that adult humans generate new hippocampal neurons. These new neurons arise from neural stem cells residing in the hippocampus. They migrate to their final positions, extend processes to connect with existing neurons, and integrate into functional circuits. The process takes several weeks from initial cell division to full integration. Several factors influence the rate of neurogenesis. Exercise powerfully promotes new neuron formation, both aerobic exercise and complex motor learning. Studies in rodents show that running can double or even triple the number of new neurons generated. Environmental enrichment also boosts neurogenesis. Exposure to novel, complex environments with opportunities for learning and social interaction increases neuron production. Stress and elevated cortisol levels, conversely, suppress neurogenesis. Certain medications affect the process. Antidepressants, particularly selective serotonin reuptake inhibitors, appear to enhance neurogenesis. Some researchers propose that this increased neurogenesis contributes to their therapeutic effects, though this remains debated. The function of these new neurons is still being investigated. They appear particularly important for certain types of learning and memory, especially pattern separation, the ability to distinguish similar but distinct experiences. New neurons may help prevent interference between similar memories. Adult neurogenesis also appears to contribute to emotional regulation. The new neurons integrate into circuits that modulate stress responses and emotional processing. Reduced neurogenesis correlates with depression and anxiety in animal models. The discovery of adult neurogenesis has profound implications. It provides another mechanism through which experience shapes the brain throughout life. It suggests new possibilities for treating conditions involving hippocampal dysfunction, such as depression, post-traumatic stress disorder, and age-related cognitive decline. While the rate of neurogenesis decreases with age, it continues even in older adults. This means the capacity for certain types of brain renewal persists across the lifespan. The aging brain remains more plastic and adaptable than previously believed. We have wandered through landscapes of neurons and synapses, traced the invisible pathways of memory and emotion, and glimpsed the remarkable machinery that generates consciousness itself. Each fact we have explored represents decades of patient research, thousands of studies, and the collective wonder of scientists seeking to understand what makes us human. Your brain, the very organ you have been using to process these words, contains approximately 86 billion neurons. Each neuron connects to thousands of others, creating networks of staggering complexity. The number of possible patterns of neural activation exceeds the number of atoms in the known universe. Yet from this biological complexity emerges something more than mechanism: consciousness, self-awareness, the felt quality of experience that philosophers call qualia. You do not merely process information. You feel, you wonder, you dream. The greatest mystery is not how the brain stores memories or regulates emotion, but how physical processes give rise to the inner world of subjective experience. As you drift towards sleep tonight, your brain will begin its nightly work, consolidating memories, clearing metabolic waste, orchestrating dreams. Tomorrow, you will wake slightly changed, your neural architecture subtly reshaped by today's experiences. In this continuous process of becoming, you are both the sculptor and the sculpture, constantly creating yourself through attention, intention, and experience. The insights we have explored tonight are not merely interesting facts. They are invitations to engage more consciously with your own mind, to nurture the organ that makes every moment of your life possible. Sleep well, knowing that even in rest, your remarkable brain is hard at work, weaving today's experiences into the tapestry of who you are becoming. Good night.